Dear students, today we are going to study about callus culture and somatic embryogenesis. Let us start with callus culture. Callus in plant tissue culture is defined as an unorganized proliferative mass of cells produced from isolated plant cells, tissues or organs growing aseptically on solid media under controlled experimental conditions. Callus-like tissue is also found to form naturally in various parts of intact plants stimulated due to deep wound or some diseases caused by Agrobacterium tumefaciens, Syncytium endobioticum, some viruses or insects, etc. And such callus-like outgrowth is known as gall or tumor. But the callus in tissue culture is produced experimentally when explants cultured under the influence of exogenously supplied hormones. Callus induction and multiplication. Let us now see callus induction. The most suitable explants for successful callus formation are often young tissues of one or a few cell types like the pith cells of young stem. Callus induction on explants in vitro generally initiate from peripheral layers as a result of wounding and in response to growth regulators either endogenous or exogenously supplied in the medium. Callus formation from explants initially involves dedifferentiation both in morphology and metabolism, and as a result callus is usually composed of unspecialized parenchyma cells and the cells lose their ability to photosynthesize. The callus growth within a plant species is dependent on various factors such as the original position of the explant within the plant, the season of the year, growth conditions of the donor plant, the age and physiological state of the parent plant, and the culture medium and conditions, etc. The presence of auxin at a moderate to high concentration and a low concentration of cytokinin in the medium promotes the formation of callus, and callus culture is often performed in the dark as light can encourage differentiation of the callus. Callus tissue from different plant species may be different in structure and growth habit and may be white or colored, soft or watery or hard, and friable, that is easy to separate into cells or compact. Let us now see callus multiplication. Callus may be serially subcultured and grown for extended periods, but the composition and structure of the callus tissue may change with time as certain cells are favored by the medium and come to dominate the culture. Actively growing callus can be maintained on culture media with an even physiological valence of cytokinin and auxin. The callus biomass usually increases 2 to 4 times after 2 to 4 weeks of growth, after which the callus can be divided and placed on fresh medium for callus multiplication. And the multiplication process can be repeated several times, up to 8 sequential transfers, before gross chromosome instability or contamination occurs. During long-term culture of Callus cultures from some plant species, the culture may lose the requirement for auxin and or cytokinin, and this process is known as habituation. Let us now see differentiation and plant regeneration. Usually, callus cells proliferate without differentiating, but eventually differentiation occurs within the tissue mass, and the extent of overall differentiation usually depends on the hormone balance of the support medium and the physiological state of the tissue. Multiplied callus can be stimulated to form shoots by increasing the cytokinin concentration and decreasing the auxin content of the culture medium, and shoot masses can be cut apart and transferred to rooting medium to obtain complete plantlets. Next, we are going to take up application of callus culture. First, callus tissue can be used to regenerate whole plants by inducing organogenesis or somatic embryogenesis through manipulation of the nutrient and hormonal constituents in the culture medium. The next application is callus tissue can be induced to regenerate genetically variable plants, since cells of the callus tissue is a good source of genetic or karyotypic variability. The third application is callus cultures are used to initiate cell suspension cultures. Next, callus cultures may serve as useful alternative sources to obtain commercially important secondary metabolites, since the secondary metabolites of a medicinally important plant can be directly extracted from the callus tissue grown from explants of the plant without sacrificing the whole plant. The next application is several biochemical assays can also be performed from callus cultures. Next, let us see the limitations of callus culture. The most serious drawback in the use of callus cultures is in the micropropagation due to the genetic instability of the callus cells. Plantlets regenerated from cells, either derived from cell suspension or isolated protoplasts, constitute unique cases of cloning called clones or protoclones, and such clones commonly exhibit somaclonal variations. Moreover, cultures in which calli are produced are also not favored, as the initial plant regeneration capacity of the tissues may decline with the passage of time. Let us now take up somatic embryogenesis. The process of embryo development is called embryogenesis, and somatic embryogenesis is the development of embryo-like structure in culture from the sporophytic or somatic cells of plants under suitable culture conditions in vitro. Somatic embryos are bipolar structures with radicle and plumule, in contrast to monopolar shoot bud obtained from organogenesis with only plumular end. Somatic embryogenesis involves three distinct steps, which are: first step is induction. It is the initiative phase where somatic cells are induced to divide and differentiate into groups of meristematic cells called embryogenic clumps. The second step is maturation. In this phase, somatic embryos develop into mature embryos by differentiating, and the mature embryo here undergoes biochemical changes to acquire hardiness. The third step is the conversion. In this step, embryos germinate to produce seedlings. Next, let us see patterns of embryogenesis. Two general patterns of somatic embryogenesis or embryogenesis in vitro are identified. They are direct embryogenesis and indirect embryogenesis. First, let us see direct embryogenesis. It refers to the development of an embryo directly from the original explant tissue without an intervening callus phase. This occurs through pre-embryogenic determined cells or PEDC, where the cells are committed to embryonic development, and such cells are commonly found in embryonic tissues. The second pattern is indirect embryogenesis. It is the formation of embryos from cells of callus tissue called the embryogenically determined cells, which are induced to form embryos and are known as induced embryogenic determined cells. The somatic embryos regenerating from explants or callus are termed as primary somatic embryos. Somatic embryos may also regenerate from tissues of other somatic embryos or parts of germinating somatic embryos and are called secondary somatic embryos. Now let us see the stages in development of somatic embryos. Somatic embryos generally originate from single cells and they develop into somatic embryos following four distinct stages. The first stage is the globular stage. In this stage, somatic embryos arise from a single cell which divide to form a group of meristematic cells, and these cells become isolated by breaking cytoplasmic connections with the other cells around it and subsequently by cutinization of the outer walls of this differentiating cell mass. It increases in size by further division to form a spherical shape structure known as the globular stage. At this stage, the primary meristem, that is the protoderm, ground meristem and procambium becomes visible. The second stage is the heart-shaped stage. This is the next stage in which the meristematic mass of cell continue to divide and differentiate with initiation of cotyledon primordia and transform into a heart-shaped embryo. The third stage is the torpedo stage. The embryo grows further, developing the cotyledon and attains the torpedo shape structure in this stage. The last stage is the cotyledonary stage. The growth of the embryo in this stage involves the division of cells inside the cotyledonary ring to form shoot and root apical meristem, and differentiation of procambium takes place, leading to the production of a bipolar structure containing root or shoot axis, that is the radicle or plumule, with a closed, independent vascular system. Let us now see factors affecting somatic embryogenesis. The first factor affecting somatic embryogenesis is growth regulators. The presence of auxin, generally 2,4-dichlorophenolacetoic acid or 2,4-D in short, in the medium is essential for induction of embryogenic clumps, and maturation is achieved by culturing somatic embryos on medium containing some doses of cytokinin, high sucrose and abscisic acid or ABA. Silver nitrate is sometimes added to the medium as inhibitor of ethylene for plant regeneration, since ethylene inhibits both somatic embryogenesis and organogenesis. The next factor is sucrose. The presence of high sucrose in the medium is required for maturation of somatic embryos to make them more sturdy and hardy. The third factor affecting somatic embryogenesis is the nitrogen source. Ammonium ion form of nitrogen is essential for induction of somatic embryogenesis, while nitrate form is required during maturation phase. The fourth factor is the genotype of explant. Explant genotype may also determine the potential of regeneration of somatic embryos in cultures. Finally, let us see the other factors affecting somatic embryogenesis. Some polyamines like putrescine, spermidine, spermine, etc. are required for somatic embryogenesis. High potassium ion levels and low dissolved oxygen levels also promote somatic embryo regeneration in some species. Let us now see the applications of somatic embryogenesis. The first application is somatic embryos can be used for synthesis of synthetic seeds or as the source of regenerable protoplast system. Next, somaclonal variants can be obtained by regenerating plants by indirect organogenesis involving Callus proliferation and differentiation of embryogenic cells from the callus. The next application is somatic embryogenesis may be used for the rapid propagation of economically important plants by multiplying germplasm that is initially present as embryonic material. Next, somatic embryos have been used for elimination of viruses in certain plant species. Finally, somatic embryos may also serve as the source tissue for genetic transformation. Let us now see limitations of somatic embryogenesis. Somatic embryo maturation and conversion is difficult in many species and is critical to the practical utilization of somatic embryogenesis. The next limitation is occurrence of somaclonal variations in indirect somatic embryogenesis may limit its application in micropropagation. Next, somatic embryo quality is often poor and field conversion frequencies are generally low. Large-scale production of somatic embryos is difficult. Now we have come to the conclusions. Plants develop callus or undifferentiated cell mass after exposure to suitable growth conditions in vitro. Callus cultures are extremely important in plant biotechnology, and development of shoots, roots or somatic embryos can be induced from the callus tissue by manipulating the ratio of auxin to cytokinin in the medium, which can subsequently lead to the formation of whole plants. Callus cultures can also be used to initiate cell suspensions, which are used in a variety of ways in plant tissue culture. Somatic embryos are formed from a single somatic cell or group of somatic cells cultured in vitro on media containing plant growth regulators that induce somatic embryogenesis. Somatic embryogenesis has also served as a model to understand the event during plant embryogenesis processes as well as a component to important plant biotechnological advancement.